My name is Allyson Berent, and I'm the Chief Science Officer for FAST. And I'm going to be speaking to you today about FAST Roadmap to a Cure 2.0. So, as I said, my name is Allyson, for those of you I haven't met, and I'm really excited to be able to get to know those of you this whole weekend. I am the CSO for FAST. I'm also the co-director of the Angelman Syndrome Biomarker and Outcome Measure Consortium, which is a pre-competitive consortium that really works hard with industry partners, Angelman Syndrome Foundation, academic researchers, and clinicians to develop endpoints that are both meaningful and measurable to our kids in clinical trials. And we had an amazing meeting yesterday, which was a scientist-to-scientist meeting that wasn't for families, but it was incredibly rigorous. We had the FDA there, and it was really awe-inspiring of how far we've come and how this is going to allow clinical trials to advance much more rapidly over the coming years. And so we've really come a long way. I'm also the co-director of InSync, as John just mentioned, which has been an incredible initiative that has been really fun, and we've learned so much from other disease organizations as well as from other experts in the field that don't work on Angelman at all, and have really come to the table to help support any gaps we have in our research and our ecosystem. I'm the Chief Development Officer at a company called Modification Therapeutics, where I help advance drugs from human candidate to human clinical trial, mostly focused on all of the non-clinical work, which we'll talk about today. And I am a veterinarian. I'm an internist, and I've been working in medical device development for my whole career. And I've been mostly working in the area of clinical trials with medical devices and animals. So I am a clinician, but I am a clinician of amazing dogs and cats. And the most important job that I have is I'm the mother to 3 beautiful little girls, and my middle daughter, Quincy, she lives with Angelman syndrome. So in disclosure, I'm the co-founder and former chief operating officer of a company that FAST started called Genetics Biotherapeutics. I'm a consultant for Ultragenyx Pharmaceuticals, and I consult and advise nearly every single drug development initiative in Angelman syndrome, mostly in the capacity of volunteer, but I will support any initiative that could benefit individuals with Angelman syndrome. I'm also a member of the N of 1 collaborative for other disorders that have much less incidence and are much more rare. So let's just start with an introduction to Angelman, and I'm going to start with the basics. For all of you newly diagnosed parents, or less newly diagnosed, but you look forward to this talk every year to remind yourself what Angelman is. So I'm going to set the stage for the next 2 days, because if you don't understand the next 5 slides, it's going to be really hard for you to follow the science of the next 2 days. So I'm going to take this slow. I want you to open your pamphlets, and I want you to follow along, because it's really important that the families understand the genetics behind Angelman. And the reason, just like John just said, is because we have an incredible, unique genetic component to this disorder that makes it prime for intervention. And you need to understand all of those aspects in order to understand the different modalities and the different ways we can approach this. And frankly, the different ways that we can fix this problem. And those of us in the room that live with this disease know that this is a major problem that needs to be fixed. And so we're going to really walk through how we can do that in a genetic, scientific way. And so I want you to follow along, because this is going to drive how you understand everything going forward. So in humans, there are 46 chromosomes. We have 23 pairs. We have 1 copy from mom and 1 copy from dad. And so 23 come from mom, the egg, and 23 come from dad, the sperm. And then together that makes 46. And Angelman syndrome is on 1 chromosome, is caused by a defect in 1 chromosome, and that's chromosome 15. Chromosome 15 has many, 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 many genes on it. But one of those genes on chromosome 15 is a gene called UBE3A. And that gene is the causative reason our children live with Angelman syndrome. It affects quite a large number of individuals, even though it's a disease that's considered quite rare. So it is orphan, which means it's rare, which means it affects less than 200,000 individuals in the United States. It theoretically could affect 500,000 to 1 million people in the world. So it's rare, but it's commonly rare, in that it's not the most rare, it's not nano-rare. The estimates are an incidence of about 1 in 15,000, Some people might say it's 1 in 8,000. Other people might say it's 1 in 50,000. We don't have the data to know how common it is, but we're working really hard through things like search and rescue and through newborn screening to truly understand how many people around the world are living with this disorder. It's generally random and not inherited, though there are rare cases of inheritance. So there are some families who have more than one child with Angelman syndrome. So it can be a situation where the mother is a carrier for a mutation. But 95+% of the time, probably 99% of the time, it's completely random. It happens in the egg of the mother when she is developing in her mother, which means that it was a random event that did not happen during your pregnancy. It was way before one of your eggs just had this happenstance, and that is the egg that actually got fertilized. So this isn't something when we think about what did I do when I was pregnant to cause this, that's not what happened. And so this is something that is just very random, and it happened to be the egg that got fertilized. And every chromosome is made up of many, many, many different genes, 20,000 plus in the genome, and therefore one of those genes is called UBE3A. And so what does that look like? Let's look at chromosome 15 a little more carefully. And if we make a schematic of that, what's really unique, and this is where I want you to pay attention, what's really unique about Angelman is that we have that one copy from mom, and we have the one copy from dad. But what's unique about this is that in neurons of the brain, not in every other cell of the body, but specifically in neurons of the brain, the father's copy is naturally silenced by something called the UBE3A antisense transcript. So the UBE3A ATS. And that is a long piece of RNA that is attached to the DNA and ultimately results in a stop. So it prevents the UBE3A gene from being read on the father's copy. But in you and I, that's okay, because we have it being read on the mother's copy. But our kids living with Angelman syndrome don't. It's either mutated, or it's deleted, or in rare cases, there's 2 silent copies, both from dad, where the dad's copy got duplicated, and now you have actually 2 silencing. So if we don't have the maternal copy being read, then we have the symptoms of Angelman syndrome. But because the imprinting, that silencing only happens in neurons, the rest of the body has a copy being read from the dad's side. So the rest of the cells of the body have 50% of UBE3A being expressed. The neurons have no UBE3A being expressed, which is why this really isn't a systemic disorder. It's a neurologic disorder, because their brain is deficient, their neurons, the cells of the brain are deficient of UBE3A. So I hope that makes a lot of sense, because what can happen from that is that we get the different genotypes, or the different types of Angelman syndrome. And so the most common type is a deletion. A deletion is where a big chunk of the mother's side is actually missing. And it's missing a bunch of genes around it, but the most causative gene is UBE3A. Oh, sorry. The second is a mutation. So that's about 10% of our population. And a mutation is where one single letter, a very unfortunate letter, got misspelled. And when it's misspelled, the gene cannot be translated to a protein that can be used in the body. And so that can be a major problem. One single misspelling can be a major clinical problem. Those with a deletion don't have one misspelling. They actually have 5 million base pairs or more, approximately, missing. So if you can only imagine 1 versus 5 million, that is profoundly different. So when we talk about deletion individuals looking different than non-deletion individuals, of course they do. They are 5 million times more affected. But when you look at them clinically, they're actually not all that different when you compare them to other genotypes. Why? Because UBE3A is the commonality, and that is likely the underlying major cause of the disorder. And that's really important to understand. And then, like I said earlier, you have UPD and ICD, which are where you essentially have two silent copies. They function very similarly in that there's not a missing mother's copy as much as a silent mother's copy in ICD or two silent father's copies in UPD. And then there's mosaicism, which is really, really rare. But the mosaic kids are so unbelievably important to this story. They have actually been the reason that we have human translation of how we can change Angelman syndrome. And what happens in mosaicism is that some of the cells are actually totally healthy and typical, and other cells have Angelman syndrome. So this happened not in the mother's egg. This happened after fertilization, when the egg and the sperm met and mom was pregnant, and one of the cells randomly got Angelman syndrome. And that one cell grew and replicated, causing many of the cells to have Angelman syndrome, but not all of them, which means they have a proportion of cells that are normal and a large proportion of cells that are abnormal. And the symptoms of those kids are profoundly different than the symptoms of the 99% of the other kids. And so this is why that is so important, and we're going to talk a little bit more about that. So what does UB3A do? So UB3A is a protein. The gene makes a protein, and that protein is responsible for all of the functions of our cells. UB3A is a very, very important protein, and so it is an enzyme that actually tags and controls other proteins. It is almost like a police officer of the neuron, and it can tell certain proteins to stick around a little longer, and it could tell other proteins, we're done with you, you're old, it's time to break you down. And if we don't have that director in the neuron, we have a lot of accumulation of old bad protein, and we don't have enough of some of the good protein. And so what happens is that tagging doesn't happen, and those proteins accumulate, and they're really, really important in the junction between the two neurons. So neurons are brain cells, and the way that our brain tells our muscles to work is that two neurons communicate together. So there's always two of them. There's the starting one and there's the ending one. And the ending one then communicates to a muscle. And then the muscle performs the function. So if you don't have two neurons communicating well, and that junction or that space between them for one neuron to tell another neuron to work, that area is called the synapse. And that space, which is sitting right here, you can see all of these little chemicals. Those are neurotransmitters, or chemicals that allow communication to happen in these neurons. And if you don't have that communication, then the neurons don't function properly. And that's called a synaptopathy. So that may be a word you read or you hear. That's what that means, is that junction's not working well. And what's really important to understand is that with Angelman, we have an accumulation of some proteins, and we don't have enough degradation of others. And so therefore, what's present at the synapse are excitatory signals. And those excitatory signals overtake the inhibitory signals that the brain needs to be calm. And so if you don't have calmness in the brain, then what happens? It's very hard for the neurons to communicate when there's a really, really strong rainstorm and all you're seeing is the windshield wipers and you're trying to drive. That's what our kids' brains feel like. There's so much going on, and they can't get all of the excitement out of the way to be in a calm space. So what happens is that there's a lot of noise. And that noise is not drowned out by inhibition. And that inhibition is called tonic inhibition. And so if we don't have inhibitory signals, and we only have excitatory signals, what happens? Seizures, lack of sleep, incoordination, inability to focus. It's really hard to listen to you because there's a rainstorm going on and I'm trying to put on the windshield wipers. I can't listen to what you're telling me because there's too much going on. So this is why our kids behave the way they behave. And so if we can clean that up and we can allow it to be calm, then they may have less symptoms of Angelman syndrome. And so I don't need to tell this audience what Angelman syndrome looks like. But I do need to tell people in the room that don't live with it that it's impactful. the #1 symptom is a universal lack of speech. That is the thing that is the hardest for a parent. There are so many other symptoms. I would say we have many disease concept models and many surveys that you all have done to share with the world what matters to you. And overwhelmingly, the lack of speech is #1. That's followed probably by sleep issues. If your kid sleeps 2 or 3 hours a night, if you have a newborn for your whole life, it's exhausting. And the impact that Angelman syndrome has on families is pretty profound. The inability to maintain employment, anxiety and depression, a complete and utter lack of sleep and productivity, social isolation. I mean, the list goes on and on. Because if you can only imagine having a newborn that doesn't sleep your whole life, it makes it very hard to function. And the most important thing is that individuals with Angelman syndrome without a transformative treatment will never, ever live an independent life, regardless of your genotype. And that's a fact. And that's really hard for parents, because we all want our kids to grow up and go to college. We all want them to live independently. We don't want them to be reliant on adults for the rest of their life. And that's what we have in front of us without transformative treatments. And that's a fact. So let's just talk a little bit about the deletion genotype, because I think this is very important for a talk that we're going to have next. For you to understand this, it's important to see that the deletion genotype has a lot of, a big range of things that are missing. So we all know that UBE3A is the most important, but there's all of these genes outside of UBE3A that are also missing in individuals with a deletion. What we don't know is the impact that those genes have on the symptoms of Angelman, because, like I said earlier, mutation and deletion act very similarly, but deletion are generally more severe in the impact of their symptoms. And so what role do all these other genes play, and how can we think about upregulating them or replacing them? And that's work that has been ongoing, that's continuing on, that we're going to talk about. When we look at those genes, there's data out there to say that some of these genes are more important than others. And I'm going to leave that to some others to talk about today, but we do want to think about that as we really have to determine how can we replace these or upregulate what exists. I want to go back to mosaicism, because we are so grateful for those kids and those families. Why? An individual with mosaicism of 1 to 5% of functional UBE3A in their whole body, which means maybe that's also in their brain. They have few to no seizures, are ambulatory, and have some speech. 20% of UBE3A, no seizures, and can speak in sentences. And over 40% of UBE3A, some of them are asymptomatic. We don't really document them when they're far above that, because they probably are asymptomatic. So what does that mean? That child is not my child. 1% would make a world of difference for 95% of our kids. 1%, 5%. What does that mean? That means the enemy of good is better. We don't have to get it perfect to give them a better life. We don't have to have 100% of neuronal transduction or gene therapy in every neuron to make their life dramatically better. If my kid could speak in sentences, if I could get here, it would be a different life that we're going to live. And so we have to work our way up, because eventually we're going to get here. But it might not be the first shot, and we have to be prepared for that. But we'll take it, because it's better, and their life will be better. So now let's talk about how we think about drug development. There's 4 pillars that we think about in order to really understand and categorize the roadmap that FAST takes to really advance drug development. The first is fixing mom's gene, or pillar 1. The second is turning on dad's gene, or pillar 2. The third is downstream targeting the synapse. Can we fix the protein problem at the synapse and calm the brain? number 3, and then what are all of the things we need to do to accelerate drug development, the tools, the models, what else do we need to do in order to ensure that we have the best clinical trials executed in the most efficient way, and that we can get drugs approved if they work, not because they fail because they have bad endpoints. And so we need to really think about all of those pillars. And just a different way of looking at it, which I find a little easier to understand, when we think about pillar 1, we're going to either replace mom's gene or turn on dad's gene. That's pillar 1 and pillar 2. We can go down to that synapse. That's pillar 3. Or pillar 4 is we need to prepare. What are all the things we need to do to be prepared for clinical trials and be prepared for drug approvals? And that's pillar 4. Okay. Gene replacement therapy. Let's talk about pillar 1. Gene replacement therapy is basically where we are going to replace a gene or replace a protein. So we're going to replace the missing gene or protein for pillar 1. And these are all of the programs currently working in pillar 1, just to give you kind of a 30,000-foot view, and then we're going to dive into each one. Pillar 2. You can do that in so many ways by activating the father's copy of that gene. We can knock down that antisense and turn on dad's copy, stop the stop. We can do that with an ASO, with a zinc finger or artificial transcription factor, with CRISPR, or with miRNA. And so these are all different ways that we can do that. And then pillar 3, downstream targeting. We have Helix and Neurin and Brown who are going to talk about their downstream approaches today. And this is where we can think about drugs that can benefit the communication between neurons. Okay, let's start with pillar 1. Replace the missing gene or protein. Adeno-associated virus gene therapy is where a virus, we're going to hear a lot about this tomorrow. This is where a virus that is a healthy virus, that all the bad parts have been taken out and it just serves as a vehicle, is going to actually carry, is going to carry UBE3A in the virus, and that's going to be carried into the neuron, and it's going to drop off UBE3A inside the neuron. And now that can be made because it's missing, and we can just replace the missing copy or the not functional copy of the gene. Lentivirus hematopoietic stem cell gene therapy, you're going to hear a talk from Jennifer Panagoulias tomorrow on this, autologous ex vivo gene therapy. This is another approach of gene therapy where we're actually going to take the missing gene, we're going to actually put it in the bone marrow, and then we're going to actually put that into into our blood, and it's going to circulate in our blood, and those cells can cross the blood-brain barrier and deliver UBE3A. And those function here. And then they can be secreted into neurons and into the area around the neurons where we know we actually need UBE3A in the cell and outside of the cell. And it can function in that regard. And then enzyme replacement therapy, where we can actually make the UBE3A protein and put it into the brain and replace the protein without replacing the gene and give them a protein replacement therapy. And that looks something like this. So enzyme replacement therapy is where we would take the protein and we would put it back in the brain. Whether or not we can modify that to cross the blood-brain barrier and go IV and put it in the brain, or whether we actually need to think about, as we've talked about many years past, putting a port into the fluid of the brain and delivering the enzyme through that port. And this has been tried and true for other disorders with incredible safety history to be able to actually just put the enzyme in. The question is, with the UBE3A enzyme, can we ensure that that can be taken up by neurons and functional? And the animal data so far would support that it can be. And so this is a strategy that we really need to think about. This is just an example of what an animal model can look like before and after gene therapy. So you could look at the mouse on the left. That little guy has Angelman syndrome. He's having a hard time. He can't keep up, a little uncoordinated. Thank goodness for that backstop, or he'd have his tail caught in the back of that thing. Poor little guy. And then after gene therapy, it's a different mouse. But it's the same mouse. So this is what it can look like after gene therapy. Our kids are not mice. But that's the type of proof of concept we need to say there is optimism for a potential path forward for gene therapy for Angelman syndrome. Let's talk about pillar 2, activating the father's copy, unsilencing the silence or stopping the stop. So like I said, there's many ways in which we can do that. Back in 2011, University of North Carolina, they published topoisomerase inhibitors, which is like a chemotherapeutic drug, but they really changed the field in showing that you can actually unsilence this and express UBE3A. So that was Ben Philpott's lab, and he did incredible work showing that you can unsilence. And then from there, Art Baudet's group showed that with an ASO or an antisense oligonucleotide, you can actually rescue phenotype by knocking it down. And then Dave Siegel's group showed that with artificial transcription factors, you can rest phenotype by knocking it down. And then other ways of doing RNA interference, like miRNA or shRNA, and then CRISPR, Mark Zilka's group, Jim Wilson's group, and Yangwei Zhang's group, all showed with CRISPR you can actually be quite beneficial by knocking it down. And so there's a lot of proof here in animals that you can make a big difference. So let's just dive in a little bit. What I want you to remember is that black is bad and red is good. And so what you can see here is that this is a mouse model. This is from Art Baudet's group. This is a mouse, the Angelman syndrome mouse model with before an ASO, where you can see that this is a control where there's no red, which means the UBE3A gene in the Angelman mouse is not lighting up. And this is after an ASO, where you can see the entire brain is lighting up. So that shows that you can activate the UBE3A gene by unsilencing the paternal copy. And that could be done with an ASO. And they showed that you can rescue some cognitive behaviors in the adult mouse model. And more recently, they showed that you can actually rescue the EEG and some of the sleep phenotype in the adult mouse model of Angelman syndrome. So this gave a lot of optimism that age may not matter as much as some used to believe, and an ASO could have profound benefit. Artificial transcription factors, this is Dave Siegel's work that he's going to talk about, but basically what that is another way of activating the father's copy of the gene, and this is new work that was just recently published, and what I just want to highlight is, remember I said that white is bad and red is good. Well, we'll use brown here, but this is typical wild type normal mouse, Angelman, and after a zinc finger. So you can see that we can activate the gene. And then he could show behavioral rescue of many different phenotypes when he did that. And so that's really exciting, showing that there is a path forward. CRISPR, same idea, but CRISPR is where we're actually going to gene edit. So this is a permanent way that you can actually knock down the antisense transcript. And a lot of work has been done showing that you can rescue phenotype. And what Yangwei Zhang's group, we're going to talk about this tomorrow, from Yale just showed was that you can actually rescue profound behavior in newborn and the older adolescent adult model. That you can actually see incredible rescue in the older ages, which is very exciting. And this is what it looks like. This is a wild-type mouse, an Angelman mouse, and a mouse after CRISPR. And what I want you to see is that that mouse is doing a better job than that mouse. So CRISPR gave this mouse superpowers. Look how great that guy looks. So this is the proof of concept we need to make decisions to move forward. And this was really exciting. Pillar 3, again, we're going to talk about the synapse. And Nurin will be speaking to us tomorrow about their compound in the clinic, and Brown will be speaking to us later today about different ways to address the synapse as well. Okay, so the last part about the animal work, and then we'll get to the good stuff. So what I want everyone to pay attention to, and this is really to John's point in John's talk, is that if you look to the left here, these are all of the different mouse behaviors that people test. Like, holy smokes, No one even tests my kid like that on the Bailey. Like, that's a lot. But all of these behaviors in a mouse are meant to translate to some of the behaviors in humans to see if you can rescue it. Cognition, motor, fine motor, coordination, learning, memory, all of these seizures, these types of activities. And what I want you to look at is that we have pillar 1 here, gene therapy, AAV or HSC, and then we have pillar 2 here, ASO, CRISPR, and zinc fingers. And what I want you to appreciate with pillar 1 is this heat map. Where there's green, there was rescue. Where there's red, there was no rescue. What I also want you to appreciate is the age. Okay? So with AAV gene therapy in adults, there's a lot of green. In juvenile, there's a lot of green. In birth, there's also green. There's still some reds, but they're contradicting, right? Same age, but different rescue. So we have to take mouse data with a grain of salt because it's just mouse data, and in different labs, the mouse perform in different ways. And so it's not perfect, but it gives you some idea. If you look here with ASO, CRISPR, and zinc finger, I want you to appreciate that the different ages again, right? Birth, juvenile, adult. You can see here, we have a lot of green. We have a lot of green. We have a bit of red, but we have some green. And so what does this tell us? It tells us that it's a splattering, but there's some crazy benefits seen in these mice at older ages. And so if anyone stands up and tries to tell us that the mouse shows us age matters, I'll have to argue with you. The only people that are going to show us that age matters are humans. Because there's incredible hope that in different experiments, older mice can do incredible things. And the major experiment we need is to show that older humans can also do incredible things. And no mouse work is ever going to be able to prove otherwise. And I want us to take that home because we have to draw a line between how much a mouse can tell us about human brain development. The human brain and the mouse brain are not even close to the same. And so we have to move on when we have proof of concept that there could be benefit with all of these approaches. There's a lot more green on this screen than there is red. And I think we should have optimism for that. A lot more green. And then finally, pillar 4. So pillar 4 is really, really important, even though it's not the drug development. Pillar 4 is what we have to do to prepare for patient access in clinical trials, for these faces that are waiting. Impatiently, they are waiting. So efforts to support clinical trial readiness for these innovative therapies are developing animal models, natural history study, the Global Angelman Registry, which I want to make an announcement and a plea. If you go outside and register, it'll take you 1 minute. You will be identified and known in the world as a child that has Angelman syndrome, which allows us to tell pharma where in the world to find you. You also get a little cozy. So go outside to the booth and register. It'll take you 1 minute. You need to be identified in the Search and Rescue Initiative to understand where you are so that we can ensure that you are heard and seen, and that when clinical trials start, we know where the population is. In addition, newborn screening efforts, which we heard a lot about yesterday, the Angelman Syndrome Foundation has put a ton of work into the Ladder Learning Network, and this is really important because this information of the natural history study and clinical trials can all potentially go into this network, this database, where it can be utilized in order for us to have comparator groups for clinical trials. And so this is incredible work that the ASF has really spent a ton of time and money on, and I think we should all give them a round of applause for that effort because it's unbelievable. And it's really helping to support how we can really pull data. But the only way we can pull data is if we have the data in there. So pharmaceutical companies, we must encourage you to put your data in there so that everybody can use the data that our kids are giving to your trials. I can't plead that enough. In addition, we have developed clinical trial training centers so that we can train the next generation of clinical trialists. We're not at a shortage of drugs. We're at a shortage of people that know how to deliver these drugs that are quite futuristic, that are going to be complicated, and that are going to require hundreds of patients to be enrolled. And we can't be working off of just small numbers of sites around the world. We're going to have to grow this network to run clinical trials. And this is something that we're very committed to do. And so we're working very hard on that. And then, of course, the Angelman Syndrome Biomarker and Outcome Measure Consortium, which we've been working incredibly hard on and we'll talk a little bit more about. So I'm not going to give this away. I'm going to give this to Wen-Han Tan for his talk to you later. But the Angelman Syndrome Natural History Study has been around since 2006, and they collect incredible data on our kids. And this is now not only in the US, Canada, the UK, Latin America, Spain. It's now happening all over the world. And these natural history studies are allowing us to pull data from well over 600 patients around the world, which is so important for regulators and pharmaceutical companies. So I cannot encourage you enough to enroll in these studies. And what's also important is that you have to be able to see what they're doing with your data, right? Just because you go and show up, but if you don't know what they're doing with your data, it doesn't feel like it has value to you. I can't explain the value it has, and I'm going to let Wen-Han do a great job of that. But there have been over 600 patients enrolled throughout the world, over 30 publications from the group in the United States, over 5 manuscripts that are still in preparation. I mean, the amount of work that has gone into this is actually profound. And we need to really encourage the community to participate, because this is what's going to help us get through clinical trials faster. The Angelman Syndrome Biomarker and Outcome Measure Consortium is a pre-competitive consortium that I already mentioned, and we had a big meeting yesterday that includes people from academics, pharmaceutical companies, various different committee members, and it was started in 2016 as a collaboration with FAST and the ASF. And it still is exactly that, but we've grown quite a bit. We went from 25 members to over 325 members. So we have done such an amazing job as a community in growing this initiative, and now this is a model for dozens of other neurodevelopmental disorders. They're all trying to create a bomb. We have an F bomb, an A bomb. There's a lot of bombs now, actually. And all of that is because of the work that this community has done to get this to be so successful. So congratulations to everyone who has done so much for this. And the tools that we have to develop in order to do this as well, we have a rat model, a pig model, a large deletion 5 megabase mouse model that we're going to hear about, human iPSC cell lines, landing pads of the large deletion UPD and ICD to understand what these genes do and how they function and which are important, organoids, and reporter cell lines, human reporter cell lines, that we can put drugs in and see if they turn on in a high throughput way to help drug companies develop their drugs. We don't want them wasting years developing the tools that we've already developed for them. So we make all these tools totally and globally available to everybody to use so that their drug development efforts are expedited. And that's incredibly important. InSync, we already talked about, which is this incredible group of key opinion leaders around the world in every area of drug development, from basic neuroscience to regulatory to industry, and they do a gap analysis on Angelman and really understand how can we accelerate from preclinical to clinical trials in Angelman. And thanks to the Simons Foundation for their collaboration on that, because it's been an incredible initiative and we have learned so much. And the good thing is we're also teaching others so much. And we have to allow that rising tides have to lift all boats. And Angelman is the rising tide, and we will lift all boats because there's 7,000 rare diseases out there that are learning from us, and we have an obligation and a responsibility to do it right for them too. So for the ABOM, I just want to acknowledge the amount of work that's been done and all of the different areas that are important to our kids. And so when we think about communication, fine motor, gross motor, global, independence, activities of daily living, quality of life, cognition, sleep, seizures, behavior. We take all of this into account, and what did we get from that? We can break it down into the ways that we can actually measure these in assessments that can be used in clinical trials. But what I want to show you is the unbelievable amount of work that has gone into this over the last 5 years. It's been an incredible amount of work that pharma companies and the foundations have really come together with our academic partners, particularly at RTI and Boston Children's, to be able to do all of this for our kids. What I also want to highlight is that FAST has put over $4 million into this because it's so important. It's so important, but it means you need to participate in some of these questionnaires and in some of these studies, specifically the natural history study, because this is how we get clinical trials off the ground. It's a huge part of our mission. It's a huge part of our investment. So each platform can potentially benefit a different targeted audience, and different individuals, we understand, may benefit from different approaches. There's not a one-size-fits-all here. We have to investigate all of the different ways and de-risk every platform in order to know which is going to have the best success. We're not going to know, unfortunately, until it's in humans. And so we have to make sure we can get there safely with the highest probability of benefit. The ultimate transformative treatments are going to be the most attractive for more commercialization. And that's really important. All of that will lead to more options for our family members living with AS. So I know this is the part that you all wait for, so we're getting there. So when we think about drug development, this is standard, right? This isn't necessarily for genetic therapies, but for every 15,000 drugs in development, 5,000 may go through regulatory rigor in order to get to a clinical trial, and of those, 10 may get into a clinical trial for one approval. Starting at 15,000 and getting to one. That's the basic research to preclinical space that most foundations live. That's where most foundations support research. The next phase is going through the regulatory process to get into humans, which is IND-enabling studies into a phase 1/2 exploratory clinical trial for safety and exploratory efficacy. Then it's the pivotal trial in order to see if the drug is working enough for an approval. So this is like the 3 main steps when we think about drug development that you need to understand. What's really important to know is what that costs. The cheap part is up here. Up to $500,000 to $2 million of research. This is for one program, for one platform, for one candidate. You could be looking around $1 million. Then the IND-enabling studies, that can range from $5 to $20 million for one candidate. Then to get through a phase 1/2, you can be looking at $10 to $50 million. And then a phase 3, but $50 to $200 million. And then just to file for the FDA to read your package is a couple $1 million. This is for one drug, guys. One. We have 24. This can take 3 to 10 years, 2 to 3 years, 2 to 5 years, another 2 to 5 years, another 1 to 2 years. This is why it takes a decade to develop drugs. It's expensive, and you have to be careful. You can't do it too fast, but there are reasons it sometimes goes too slow. And so this is where we were when I came into the community when my daughter was diagnosed. This is what I was looking at 8 years ago. There was an AAV gene therapy, thanks to Ed Weber. There was an ASO, thanks to Art Baudet. There was artificial transcription factors, thanks to Dave Siegel. And there was a bunch of downstream targets that were being explored, one of which thanks to Ovid Therapeutics. 2 years later, because we had the largest infusion of funds to FAST of $5.8 million by a single family touched by Angelman syndrome, we went to 15 programs in 2 years. of which 10 of them were funded by FAST because of that infusion of funds. So you can see all of these. 2 years ago, we were at 34 programs. All of us getting closer and closer to clinical trials and IND. 3 ASOs in the clinic, multiple gene therapies and IND-enabling studies, downstream targeting, heading to the clinic. And then this happened in May of 2022. Biotech lost tons of funding, and when Angelman was on the early pipeline, they fell off the pipeline because people needed to change their priorities in order to keep the doors open of their companies. And all of these early biotechs and these early rare disease companies were dropping programs. And Angelman syndrome was dropped. It was dropped by a lot of people. 34 programs, we lost 13 in the last 2 years. The chutes and ladders of drug development are real, and we are going to gain and lose programs left and right. And if we're not ready for that, we're in the wrong space. There are going to be clinical trials that fail. They may fail because the drug doesn't work. They may fail because we have bad endpoints. But over my dead body, will it be because we have bad endpoints? But we have to be prepared that early discovery programs are not going to make it to the clinic. They're going to fail in animal models, they're going to fail in toxicology models, or they're not going to get funded. And they're going to fail on the line because no one funded them. This is what happens in drug development. But today, we have 23 programs, 13 of which are funded by FAST, and I can assure you, the only way FAST will ever stop a program is because we can't afford it, or the science tells us it's not the right thing to advance. And that will happen, but we will never stop a program because we changed our priorities, because our priorities will absolutely never change, which is why we will continue to advance the programs we invest in. We will continue to support anybody in partnership that will advance the programs we invest in, because our kids are waiting for this, and our priorities just can't change. So right now, this is where we are today. We have 4 programs in the clinic. And we have a bunch of programs approaching or getting near IND-enabling studies, 13 of which are supported, if not wholly, hugely by FAST. This is where we were 8 years ago on Pillar 4. New models, regulatory engagement was very early, registries were starting, diagnostic efforts were super early, we didn't have much in policy, newborn screening, the disease, all of this wasn't happening. And here we are today. We have moved so far on Pillar 4, which may not be as colorful as Pillars 1, 2, and 3, but just as meaningful. Because we can't get to clinic, and we can't approve drugs if we don't have it. And so we've come a long way in a short period of time. And when we funded the Rush Pediatric Neuroscience Center, the FAST Center for Translational Research, it was meant to really ensure we raise the next generation of clinical trialists. We are not here to run holistic care clinics. The Angelman Syndrome Foundation does an amazing job at that. We do not want to compete with that in any way. We want to support that. But we also know holistic care clinics and clinical trialists that have time to be full-time running clinical trials are 2 different needs. And we need to help support that, and it's really expensive to do both. So we're here to help support that so we can ensure that all of these centers around the world can run trials safely in a huge number of kids. Because if you see where we're going in the next 5 years, we're going to need hundreds of patients in trials. And right now, that's really hard to do with the amount of centers that can accommodate enrollment. It's a problem. And so where we are here, this is Rush, and as they train fellows, we want them to disperse around the country and around the world. For the next group to train fellows, to disperse, and to disperse. Because we need to arborize the world in running clinical trials and delivering novel therapies. It's incredibly important, and it's our obligation to do it, and it's not inexpensive, and we have to lay the groundwork now. So really, our mission is to bring these transformative treatments to our global community, and we do that, as you can see, from discovery and animal models endpoints, engaging the community and knowing where they are around the world and knowing where they live so we can make sure their hospitals can deliver these drugs, and then making sure we can run clinical trials in the most efficient way. And so really, that continuum is what allowed the NIH to look at Angelman syndrome and the incredible work that Yang Wei and Zhang Bing at Yale put together to say, you guys have an infrastructure to go from benchside to bedside. We actually do in Angelman syndrome because we created it. And so we're out of the silo of lab to lab, and we can have Yale, for example, doing discovery, and Liz Berry-Kravis at Rush doing a trial, and FAST doing the in-between. And that is benchside to bedside. And the NIH saw that and gave this team $40 million 2 weeks ago to be able to do this. And I couldn't be more proud of our community, because the reason that they thought Angelman was the right disease to do it on is because of what you all funded to make that possible. The great minds in science, we didn't create, but we recruit. The great minds in clinics, we didn't create, but we can recruit, and we can bring them together. And that's what we are, is we're facilitators to make things happen. And so what I want you to appreciate is how expensive drug development is and how long it can take. But FAST is committed to this $15 to $25 million space. We are committed to take preclinical, go through IND enabling, and get into the clinic. Because at this point, not everyone is willing to fund that, and we will never let things stop for funding. And so in 5 years, this is what I see. I see nearly a dozen programs in the clinic with at least 2, if not 3 or more drug approvals, and tons of programs and IND-enabling studies. Numerous potential therapeutic platforms are invested in AS. AS is unique and has a very elegant gene targeting approach that we can really manipulate, promising clinical results to date that we're going to hear about tomorrow. And we're focused to identify gaps and accelerate all potential strategies so that we meet all shots on goal. Because we do not know what's going to work in these kids, but we will, for certain, not stop until we figure that out. FAST is working hard to create the infrastructure for human drug delivery, because that's what we need, and that's what our families deserve. And so, as Art Baudet always says, who is my idol? There are many disorders that will not be cured or treated in our lifetime, but Angelman syndrome will not be one of them. And I couldn't agree more. So don't be scared. Have hope and have promise that it's time for us to continue to dream big, because everything is possible. Nothing is impossible. So keep at it. Thank you.